Hello friends, this video on biomolecules part 5 is brought to you by examfear.com. No more fear from exam. Now we will look at the classification of carbohydrates. How can we classify them? Now we classify carbohydrates on the basis of their behavior on hydrolysis. Now what is hydrolysis? We are going to talk about two important processes here. One process is where I was talking about anabolic and catabolic pathways. So there is one process where you make complex structures from simple structures. There is one process where you break down complex structures to form simpler structures. So here we'll talk about two processes. There is one method rather, where bigger carbohydrates can be broken down to give smaller carbohydrates. So that process is hydrolysis. What do? We add water. So hydrolysis means cutting down with water. So you add water and it breaks down into smaller carbohydrates. That other process is condensation, where you take out water, you pull out a water molecule and a bond is formed which gives rise to a bigger carbohydrate. So these are the two processes which we will be talking about very soon. Now it has been seen that different types of carbohydrates, when hydrolyzed, they show different behavior. Some of the carbohydrates on hydrolysis, they give smaller compounds. Some of them cannot be hydrolyzed further because they are already the simplest carbohydrates. So based on their behavior, carbohydrates have been divided into the following types. Monosaccharides. Mono means one. Saccharides means sugar. So monosaccharide is like one sugar. So you cannot further hydrolyze it. Next is oligosaccharides. Oligo means few. So under oligosaccharides you have di, tri, tetra till deca. That means 2 to 10. So mono is 1. Oligo is 2 to 10. That means 2 to 10 units of sugar. And the last one is polysaccharides. Poly means many. So these are the three types of carbohydrates: monosaccharides, oligosaccharides and polysaccharides. Now we will talk about each of them gradually. So let us start our discussion with monosaccharides. So what are monosaccharides? Mono means one. So this is going to be the simplest form of carbohydrate. So carbohydrate that cannot be hydrolyzed further to give simpler unit of polyhydroxyaldehyde or ketone. So it cannot be hydrolyzed further. That means they are the simplest form of polyhydroxyaldehyde or ketone. So as I said, hydrolysis is that process where bigger or complex carbohydrates get broken down into simpler carbohydrates. So this is the process where you can say that bigger units getting broken down or getting converted into smaller units. Now monosaccharide is the simplest form, and that is why you cannot further hydrolyze it. So those carbohydrates which are the simplest form, which cannot be hydrolyzed further, are called monosaccharides. There are about 20 monosaccharides which are known to occur in nature. So some of the examples of monosaccharides are fructose, glucose, ribose, erythrose, glyceraldehyde. These are all examples of monosaccharides. We will talk about the structure and properties of some of the important monosaccharides like glucose and fructose. Now let us try to understand the processes dehydration, synthesis and hydrolysis, because as and when we talk about carbohydrates, we'll keep talking about hydrolysis. So it is very important that you understand these processes. Now first we'll talk about a process called dehydration synthesis. Now what is dehydration? The word dehydration in general means taking out water. So we often say, right, that we should protect ourselves from dehydration. That is, we should protect ourselves from loss of water. So dehydration is nothing but loss of water. So this is the process where pulling out water to build a bond between multiple sugar units. So in this process, what we do is we take monomers, that is simple sugar molecules. So we take monomers, that is simple sugar molecules, and polymers are formed. Polymers are bigger structures or complex structures. Then how do we form that? We take out water and a bond is built between the two sugar units. Let us take one example. Here you can see the structure of glucose. Now both of these are structure of glucose. This is the open structure and this is the closed structure or the cyclic structure. Now we'll get into those details later. For now, you just understand that this is the structure of glucose. Now, if you bring two glucose molecules together, what will happen? You will have one molecule like this and you will have another molecule like this. So, basically, there will be one OH bond here. Again, there will be another OH bond in the other molecule, right? So, it will be somewhat like this. Let us suppose this is the OH of the first, this is one glucose. Again, there is another glucose. This also has OH. Right? And again, here you have other structures. Now, I am not drawing the entire structure. What I am just trying to explain is when you have two glucose units combining with each other. So, what happens in the course of that process is this OH and this OH. So consider these two. So from these two, H2O is taken out. So if you take out H2O, what is left? This H will go, this is gone. So you are only left with one O. So these two glucose molecules will get connected by this bond. So instead of two glucose molecules, you will now have one sugar unit. And this bond and this entire structure will still remain the same. But now instead of two, you just have one structure. And this bond is known as a glycosidic bond. We will talk about that bond also in even more detail. But for now, you should just know this is a glycosidic bond. So this process is known as dehydration synthesis, where two sugar units or two monosaccharides come together. One water molecule is taken out and a glycosidic bond is formed. Due to the formation of that bond, a bigger sugar unit is formed, that is a polymer is formed. So in this case, this was a monosaccharide, this was a monosaccharide, but the result is a disaccharide because the result has two sugar units basically. So whenever we use the term sugar, we often use it synonymously with monosaccharides. Okay, so here this structure actually has two units of glucose. So this is a disaccharide and this process is known as dehydration synthesis. This is also known as condensation. So condensation is another term for dehydration synthesis. Please understand this process properly, because throughout this lesson, wherever you see that polymers are getting formed from monomers. Now you understand what are monomers. Monomers are always the simplest form of a biomolecule. For example, when we talk about carbohydrates, monosaccharides are the monomers. Oligosaccharides, polysaccharides, they are all polymers. So similarly, when we talk about proteins or when we talk about lipids or when we talk about nucleic acids, everywhere the polymers will be formed from monomers and they will be formed by the process of dehydration synthesis or condensation. So this process will remain the same. Take out one H2O and a bond will be formed. It is just that everywhere the type of bond which is formed will be different. So here this bond is known as glycosidic bond. That is because glyco is related to glucose. Glucose is carbohydrate. So that means it is glycosidic bond. So now if you understand this process of dehydration synthesis, then I don't really need to explain you much about hydrolysis. So hydrolysis is just the opposite of dehydration synthesis. So in this process, we break the bigger molecule or we break the polymers back into monomers. Now, a simple common sense. When you wanted to form the polymer from monomers, what did you do? You took out one water molecule and you got a disaccharide. Now, if you want to do just the opposite thing, what should you do? Exactly. You should add one water molecule. So adding that water is known as hydrolysis. So it is the reverse process of dehydration synthesis. So what do we do here? Here monomers are formed from polymers. And in order that monomers are formed, what do? We'll add water in this case. The term hydrolysis means, hydro means water, lysis means to cut. So to cut down with water. So when you add water, what gets cut down? The disaccharide. For example, this was your disaccharide. This was the glycosidic bond which was formed. Right? Now when you add water, what happens? This bond gets broken down and you again get the two glucose units. So this process is called hydrolysis. That is, with water, you are cutting down the bigger molecule into smaller molecules. So are these two processes very clear to you? Because this is very important. If you don't understand this process, you are not going to understand anything which I'll talk about in the next couple of slides. So please recap the slide, but get it into your head. Just understand it. So dehydration synthesis is condensation, where you pull out water to build a bond. And hydrolysis is adding water to break a bond. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.